Thank you very much for joining us in this Physio webinar today. This is an event organized by graduate students from the Physiology graduate program of the Institute of Biosciences of the University of São Paulo. This is our team. We organized 4 very interesting talks happening during October and November with speakers from different countries talking about different things in physiology and their intersection with other areas. Please follow us to get more information and so you do not miss any of the talks. Today, our speaker will talk about a very integrative theme in physiology, chronobiology in social insects. Professor Guy Bloch is a bachelor in biology from the University of Tel Aviv and PhD in zoology from the same institution. Currently, he is professor in the Department of Ecology, Evolution, and Behavior at the Hebrew University of Jerusalem. The main themes of interest in his research encompass evolution and the mechanisms underlying sociability and social behavior, studying bees as a model and employing integrated analysis at different biological levels, from molecular to social. In recent years, his research has been focused in the social chronobiology, the interaction between individual and social circadian rhythms, and how these aspects are related to the colony context, such as the division of labor. In addition, he has carried out studies on the social evolution of juvenile hormone signaling, interaction between sleep and social behavior, and the involvement of non-coding RNA and RNA editing in behavioral plasticity. Dr. Guy Bloch, thank you very much for your presence here today. After your talk, we'll have 30 minutes to take questions from the audience, which will be selected by our team. Everyone can ask questions through YouTube chat, and they can also be in Portuguese. We will be here in the background in case of any technical problems, so don't be afraid to ask for help if anything bad happens. And right now, I'll give the word to you. Thank you.
Thank you very much. So now you probably see my screen.
Yes.
Okay. So it is a real pleasure to be here, and thank you for the invitation and for the kind introduction. And as you mentioned, we are interested in various aspects of the interplay, basically, between physiology and social behavior, and in the ultimate sense, also the evolution of sociality and the evolution of social behavior. And we study bees as model. And today I will speak about what we call the social clock of the bee. And I will present mostly on honeybees, but we actually work in parallel on honeybees and bumblebees. I will show a little bit about the bumblebees. So this is a line of research that, as you mentioned, we pursuing for quite a time. And we were fascinated. Actually, we started to focus on the system because we were observing the bees and trying to learn about the division of labor. And we discovered that the different tasks that they take in the colony are also associated with differences in their behavioral rhythm. But we extended it to several lines of research, which I will present in a moment. But what I try to show in this opening slide is basically that we are interested in 2 directions of this interplay. One of them is the influence of the social environment on the clock of individual bees. And the other direction is how the behavioral rhythms of the clock of individual bees are interacting to form colony-level coherent behavior. And of course, these 2 lines of research actually are interactive. So the honeybee specifically was actually one of the main models in chronobiology, and specifically in studies on the behavioral aspect of circadian rhythms. It was, I think, the first model for behavioral circadian rhythms. And this was because honeybee workers, they do a lot of foraging behavior because they forage for the sake of the entire colony. So they just go in and out. And already about 100 years ago, it was discovered that they have strong rhythms, and that relates to their activity. And later it was shown that they can time their visits to flowers. And it's important because flowers actually, at least some of them, provide the reward like nectar and pollen at certain times of the day. And bees are very good in organizing their activity such that they visit flowers when they are most rewarding. And it's also important, later it was discovered that the clock is also important for sun compass orientation, which is another aspect of their behavior, and for dense communication, which all of them rely on the position of the sun, and because the sun is moving from east to west, they need to use the circadian clock to compensate for the sun movement. But as I mentioned, the focus of our lab is mostly on the interplay between social behavior or sociality and the circadian clock. We call it socio-chronobiology. It's always nice to come with some kind of a buzz name. And the five main things that we study under this title are social influences on the ontogeny of circadian rhythms. So at least in honeybees, but to a certain extent also in other social insects, the adult individuals, the adult females, the workers, they develop circadian rhythms as they age. So they start with no circadian rhythms, and then after a while, they develop very robust circadian rhythms. And this is to some extent under social regulation. I will not have time to speak about it today. Another aspect that we actually studied quite a lot is the interplay between division of labor and circadian rhythms, and I will extend on it. There is also reproduction-related plasticity in circadian rhythms, specifically for queens. Queens, when they are virgin, they have strong rhythms. When they are reproductive, they're actually active around the clock, and we did a little bit of work on it. We're still doing. The other big subject is social entrainment or social synchronization, and I will try to share some recently published and unpublished results. So this is actually in the broader field of chronobiology. This is the larger aspect of social interactions affecting the clock. It has been studied in many different organisms, mostly in rodents and other mammals, but to some extent also in insects. And finally, there is a very interesting aspect of social influences on sleep, because of course, when we started to work on plasticity and circadian rhythms, it's always linked to sleep. Sleep is influenced by the circadian clock, and we slowly get more and more interested in sleep. And again, unfortunately, today I will not be able to speak about it. So this is actually, most of the talk will be on these two topics. And to start speaking about socially regulated plasticity in circadian rhythms, which is, in this case, it's related to division of labor, it's always great to start with this beautiful picture by Dali, which shows not only plastic clocks and sleeping creatures, but also social insects. I don't know if I don't have much interaction with the audience, but I don't know if you can see the social insects, but if you will look carefully where my cursor is now, you will see that there is a clock here with many ants actually, and ants also show socially regulated plasticity in their circadian rhythms. And then the basic system that I started to work already in around 2000, when I was doing my postdoc, relates to the division of labor. So it was well known, as I mentioned before, that foragers have very strong circadian rhythms. And actually, if we observe their behavior and we try to plot it using double plot actogram, which is what we are showing here, we will see something like this. So in a double plot actogram, each row is a day And here you can see the hours of the day, and you can see that each day is double plotted. You can see that the day here is basically the same one that is presented here. This is done just to facilitate visual detection of the rhythms. And the black bar correspond to the level of activity. And you see that the foragers are active during the day, and they are not active during the night. And now we know very well that they actually have a very good night's sleep during this time. And on the other end, the young bees, as I mentioned, they don't have circadian rhythms, and typically in the hive, they perform several in-hive activity, but the most prominent one, the most common one, is caring for the brood. So brood is a general name to the pre-adult stages. Here you can see larva, and next to it you can see sealed brood, which is typically pupa. There are some also eggs that we don't see on this comp, and the bees are basically tending the brood. And if we record this activity, we see something like this. We see that the level of activity, so this is the record based on visual inspections, visual observations of an individual bees that we mark them individually with number tags. And you can see that by contrast to the foragers, she actually has no clear rhythm. She's active around the clock with no circadian rhythms. And we were really fascinated by these observations because it makes a functional sense. So we believe that foragers, of course, they need the clock. They need to know when is day and night because the plants that they are feeding on, the flowers are open during the day. And they use it for time interval visits to flowers, for sun compass navigation, and for dense communication, as I mentioned before. So they actually need a clock. On the other end, we believe that the nurse bees can provide better care for the brood when they are active around the clock. For example, they can make sure that the temperature and the other conditions in the hive are appropriate, and they provide more food and more cleaning of the larva. So, but there is still the issue that the nurse bees are typically also younger. So we didn't know if it's related to age or to task. So we performed several experiments in which we uncoupled by means of social manipulation, age and task. And the most remarkable experiment is what we call a reversion. So we induce some of the old foragers that already have they have strong rhythms with a nightly sleep to revert back and behave like young bees. We induce them to care for the brood again. And when we did it, we saw that they moved back into around the clock activity. And this was the first evidence that plasticity in circadian rhythms in any animal can work in both directions. And another way to ask and to explore the relationship between division of labor and social organization and plasticity in circadian rhythms is to use the comparative approach. So we decided to focus on bumblebees. One of the reasons is that I really like bumblebees. That's the animal that I studied for my PhD. And the other reason is that they are taxonomically related to honeybees, but their division of labor is organized differently. It's not related to age, or age has very little effect on it, but it relates to body size. The larger individuals typically forage outside, the smaller one care for the brood. And here you can see actually nurse bumblebee feeding a larva. Those are all these, what looks like small balls are actually larvae that are covered with a little bit of wax. And we did behavioral observations, and we found strong rhythms for the foragers and no rhythm for the nurses. So although it's not related to age, it's still we see the same plasticity. And we and several other groups characterize the same system in several ants in which sociality has evolved independently of bees, and basically we and them found the same plasticity. So we believe that there is a genuine link between plasticity and circadian rhythms and social organization. But however, when we collaborated with several other people. This is a follow-up of a conference that we organized in Israel, and some of you actually participated in the conference. And we found that there are quite a few animals that show comparable plasticity. For example, shark and other open sea fishes, they need to just be active all the time in order to get sufficient oxygen from the water. Marine mammals, they need to surface in order to breathe, and they are active around the clock. Many migratory birds fly during the night and are active during the day. So during the migration period, they are actually active around the clock. Some birds, like this common swift, basically for something like 10 months of the year, they are just flying all the time, 24/7. They actually stop flying only during the breeding system. And of course, many Arctic animals, during the long summer days, actually, when the sun never sets, they're also active around the clock. So similar plasticity is found throughout the animal kingdom, and it's not unique to bees, but bees provide a more tractable system, perhaps, to understand this system. So one of the first things that we did is in the context of the honeybee genome project, we characterized the clock system of the bee. When we started, there were two models: the insect model, which was basically the fruit fly, and the mammalian model was basically the mice. And the basic organization of the clock was similar, but the clock genes were a little bit different. And when we studied the honeybee, we found that it's actually different than Drosophila, and actually it's very similar to what we find in human and other mammals. So pair and the mammalian type cry form the negative loop of the circadian clock. So basically, under the control of cycle and clock, two other clock genes, they activate the transcription of the pair and cry genes. They are translated into proteins secreted to the cytoplasm, interact in some way that we don't know exactly how it's working in bees, but we know pretty well how it's working in mice. They transported back into the nuclei, in which they inhibit their own expression by acting on cycle and clock. And this is forming a negative feedback loop of about 24 hours, which is the molecular basis of circadian rhythms. As you probably know, in 2017, Rosbach, Young, and Hall, which discovered this machinery in Drosophila, were awarded the Nobel Prize in Physiology and Medicine. And there is also another loop here that is also different than in Drosophila and similar to mammals. And characterizing this system gave us the tools to better study the molecular mechanism of plasticity in circadian rhythms. So we perform a lot of experiments in which we just characterize clock gene expression in nurses and foragers. Here you see a summary of some of these studies, but the results were actually very similar. So in this case, we took the entire brain of the bee and extracted all the messenger RNA and tested it in several different hours. We did it in DD and in LD. In this experiment, it was done in LD. And as you can see, the mammalian type cry, perioid, cycle, and clockwork orange, four of the major or canonical clock genes, show very robust rhythms in foragers. Cry and pair are typically very similar in phase, and the cycle is typically about an antiphase to them. And when we look on nurses from the same colonies, we see attenuated rhythms at best, and actually in most cases there is no significant rhythm. So at least in the level of transcription of the entire brain, we see that there are differences in the machinery of nurses and foragers. And this raises a lot of interesting questions that we pursued in following years. For example, because we are interested in sociobiology, we ask what are the social signals regulating plasticity in circadian rhythms. We can also ask, why do around-the-clock active bees in a constant hive environment, because the the honeybee colony is very well thermoregulated. The hive is essentially constant, the temperature, the humidity, it's always dark. So why do they need a clock if they're anyway active around the clock? We still don't have answer to this. We can ask, do they sleep because they are active around the clock? So we already know the answer. They do sleep, but they sleep differently than foragers. And as I say, I will not have time to speak about it. And if they stay in such a constant environment, how can they be synchronized with day-night cycle? Because I will show you that they actually can synchronize. So this is actually, I hope I will be able to speak about it in the end of my talk. So to start studying the system of the social regulation, we suspected that the contact with the brood is actually the factor that is inducing nurse bees to be active around the clock. And here you see Yair Shemesh, a graduate student that led these efforts together with several other students in the lab. And what he did is that inside normal colony in which you can see the nurse bees, and here the white spots that you see here, if you see my cursor, are actually larvae. He put a piece of cage, it's made of mesh, and here we have bees that are exposed because it's made of mesh. They are exposed to the smell of the colony, including the odors of the larva, but they cannot contact the larva. And he simply performed behavioral observation. We can mark them individually with numbers that you can see here. And this is like one individual, red 47, this is white 20. And this one is a normal nurses. You can see that she is active normally during the light phase, the day, and the dark phase. This colony was maintained in a light-dark illumination regime. It will be the same in constant darkness. On the other end, the bees that are inside this broodless piece of comb, they are much more active during the day. And when we did the statistics for a pretty large sample size, we did it with 3 different colonies. The results were very clear. No rhythms for the nurses, strong rhythms with activity during the day for the bees on the broodless comb. But when we remove these bees and monitor their activity in isolation, in individual cages, both of them actually showed very strong circadian rhythms, and both of them were actually active mostly during the subjective day, because the monitoring was done in constant condition. But you can see that they have pretty nice rhythms. And when we analyze brain gene expression for some of these bees, we found, as expected, as I showed you before, very strong rhythms in pair and cry with a similar phase for the foragers, attenuated rhythms or no rhythms for the nurse bees. And what about the bees on this broodless piece of comb? So they actually showed very nice rhythms, at least for pair, it was actually comparable to the foragers. Remember that they are also younger than the foragers because they are same age as the nurses. By the way, one of the advantages of running this experiment with bees is that the queen can lay a lot of eggs every day. It can be up to 2,000. So all these bees are actually sisters. They are genetically related, so there is no much genetic variation in the system. So it's consistent with the idea that the contact with the brood is actually what regulated plasticity and circadian rhythms, and we showed it in several other experiments, including experiments in which we exposed the bees to brood in small cages. And until this moment, our model was that because the larvae need to be fed, they need to be tended around the clock, it's like babies that are crying and the nurses are responding to the signals and they come to feed the larvae. But later we tried to develop a system to analyze individual honeybees in the presence of brood. And Muki Nagari, who led this project, actually we couldn't do it with larvae, and he tried to do it with kept brood, which are mostly pupa. This is a piece of comb of a kept brood. So under each one of these covers, there is a pupa. And here you can see the entire array that we monitored. And we put an individual bee inside this cage and monitor its activity. And let's go to D and E, which are the summary of two different colonies. We repeated it with two different colonies. And we have some cages in which we have empty comb with no brood, some with brood, and some with brood that we removed. And you can see that the power of circadian rhythms, which is a proxy for the strength of circadian rhythms, was significantly reduced in the presence of brood. In the second colony that we show here in E, the difference was actually even stronger. And if we look on C, we see for the same two colonies, Mookie actually also look on the different number of sealed brood, because it was not exactly the same in each one of them. And he found a weak but statistically significant correlation for the effect of the number of brood on circadian rhythm. So it's provide even a stronger support for the idea that the contact with the brood, even brood that doesn't need to be fed, is sufficient to induce these to be active around the clock. So when we show these studies on plasticity in circadian rhythms and the fact that bees can move between and switch between activity with and without circadian rhythms, most of the chronobiologists were thinking about two possible explanations. One of them is that in the presence of brood or young bees, basically their clock is not working, it's not functioning. And the other possibility that people suggested, which is almost the opposite, is what the chronobiologists like to call masking. So the idea is basically that the clock of the nurse bees is working, but because behavior is affected by social factor, it's masking the effect of the clock. But the clock should be similar for nurses and foragers. And as I will show you in a moment, actually both hypotheses cannot fully account for the way that we currently understand the system. So to start understanding the system, Yair did an experiment in which he removed the bees from the colony at certain times during the day. So the idea is that if the removal of the bee from the colony activate its circadian clock, we will see a clear correlation between the time that we remove the bee from the hive, the nurse bee from the hive, and the onset of locomotor activity. So this is the red line that we see. That's what we expected. On the other end, if there is, the clock is normally functioning, the clock of nurse bees is normally functioning, and it's entrained with the environment, it will be their onset of locomotor activity, their daily onset, will be correlated with the time of morning. And in this hives, we could control the time of morning. It was 8 a.m. And that's what he did. He took nurses that are active around the clock in the hive based on behavioral observation, removed them to our locomotor activity monitoring system, monitor their activity, determine the onset of locomotor activity, and this is the results that he found. So clearly, it is correlated with the morning time. And this was the first evidence that actually the nurses, although they are active around the clock, they do have at least some of the internal clock are functioning. One of the other studies that we did is that we used a microarray approach. So by that time we had the honeybee genome, and Jim Robinson developed nice arrays, and we collaborated with him, and we tested the bees around the clock, and we characterized all the probes on the microarray that show circadian oscillations, and we found about 540 probes for foragers. And here you can see the time of collection. This is the subjective day, and this is the subjective night. There is like peaks in the middle of the subjective day and the subjective night. And the percentage of rhythmic probes compared to the number of probes that we have in the array is actually comparable to diurnal animals like Drosophila or mice that were studied before. And then we look specifically when we look on nurses that are active around the clock, and we confirmed it by behavioral observations, of course, we found significantly smaller number of cycling probes, but still it was quite a robust number, 160 probes, which suggests that at least some processes are under circadian regulation in the brain of nurses, as consistent with the study that I showed you before. When we look on specific pathways or specific families of genes, for example, this is for cytochrome P450. It's a very important group of enzymes. And I remind you that here we look only on the brain. We found that many of the P450 genes are actually cycling, but they show different phase in nurses and foragers. So at least some of the processes are in different phase. Other processes that are cycling in foragers are not cycling in nurses. To further extend this line of inquiry, we performed several studies we tried to characterize the clock, the neuroanatomy, and it was quite frustrating until only recently we have a very good antibody, and we could characterize the clock system in the brain of the bee. This was a collaboration with the lab of Charlotte Foster, the same individual that characterized the clock in Drosophila, great neuroanatomist. And this is basically a summary of what we found. The green spots are neurons, in this case, that express the clock gene period. This is the one to which we have a very good antibody. And in purple or magenta, you see expression of PDF. This is a very important neuropeptide that is known to function in the clock system of insects. And in the left hemisphere, you actually see summary of the main arborizations of the fibers stemming from this PDF expressing cells. And if we take a closer look on some of these clusters, we see here, for example, we see a double staining with HRP, which stains specifically neurons. Green is for period, the clock gene. And you can see that many of the pair expressing cells are actually neurons. They are marked with white arrows, but some of them, which has a different structure to the nuclei, it's more like elongated. You see here, marked with green, are actually probably glia cells. And if we look closer on this hub of the circadian network, the area of the major group, which we call it the lateral neuron 1, it's actually the brain is organized, the clock system is organized very similar to what we know in Drosophila. So this is a large group with about 70 cells in each brain hemisphere. And just next to it, there is another group, which is much smaller but larger cells, which are the LN2. And they also express PDF that you see here. And this is the double labeling, the staining. You see that these cells are much larger. And to start and understand how the plasticity in circadian rhythm is expressed in this system, we use again observations hive. We have freely foraging bees, and we collected nurses and foragers at several different time points and stained them for pair and PDF and summarize their expression level. And here you can see the summary, like representative results. And you can see that in the dorsal lateral neuron 1 and lateral neuron 2, the main clusters of clocks, there is cycling in foragers and in the nurse. And it's comparable, at least in this example. This is the LN2 with the PDF staining. And here you see one group of glia cells that we measure in the optic lobes, but we didn't have the capacity to do it properly. We will repeat it probably. And if we summarize the results from experiments with bees from two different colonies that are not related, we see strong cycling in both foragers in blue and nurses in red, in all the clusters besides the glia cells, which suggests that at least the neuronal network, the neurons that express pair in the brain, cycle very similarly in nurses and foragers. which is providing probably the ultimate support for the idea that they have functioning clock. So if we summarize what we know about nurses and foragers, in foragers, it's like other diurnal animals. They have very strong rhythms, and many different processes and clock in their body are like in other diurnal animals. If we take a young bee or an old bee and put it without a brood, basically we will see the same picture. But if we look on nurse bees, it's actually a different picture. So in this case, there are less cells that are cycling. This is marked by this red mark here. Some cells or some circadian processes have the same phase as in foragers, but others are in different phase. And this includes, for example, the antenna that we characterize their responsiveness, and we found that it's cycling in both nurses and foragers, but with a different phase. Now, because the nurse bees are in a constantly dark and well thermoregulated hive, we ask how they can be entrained to the environment outside. And we perform a set of experiments, try to answer these questions, the bottom one here. So one way to think about it is to think about the body of mammals and Drosophila, for example, and other modern organisms. So animals like Drosophila and zebrafish, which are essentially transparent, each one of their clock cells inside their body is exposed to the environment and can be easily synchronized. On the other hand, in our brain, in which the clock system, at least the central clock in the brain, in the SEN, is actually not exposed directly to light, so light information and other information about the cycling environment is perceived by the sensory system, mostly the eye, and they interact with some cells in the SEN, and the cells in the SEN, one of the ways that they can distribute this information is by means of a neuronal network. And the same system can also use hormones or other changes in the whole body environment to entrain internal organ like the kidney and the liver, for example. So if we translate this principle to the honeybee colony, one possibility, this is like the zebrafish / Drosophila model, is that the nurses can sense the condition outside. Either they're exposed to light when they approach the hive entrance, or that they go to the hive periphery. Maybe there is a kind of social network similar to the neuronal network in the SEN. You know, some bees that are exposed to the environment and train the other, and then that means a social network and train the entire colony. Or that the bees that are active actually create an oscillating environment. This is also entraining the nurse bees. There was also an idea that the queen maybe is entraining the colony. There is even a paper or two about it, but we don't think this is the case because the queen don't have rhythm, and they are the last ones to know about what is the conditions outside. So to start studying the system, we perform a lot of experiment testing the hypothesis that social synchronization is the way of entrainment, and we showed that social synchronization is very effective, even if we put a group of bees inside small cages, they can entrain each other. But then we were so impressed by the strengths of social synchronization that Ada and Rothschild, to study the system, decided to compete light entrainment and social entrainment. And the way that we did it is that we put observation hives with a colony inside the laboratory. We exposed them to a certain light-dark illumination regime. And we let the bees also forage outside, as you've just seen. Now, we did the light-dark illumination regime and the opening and closing of the hive, which determine the forager activity, 6 hours apart. So we uncoupled social synchronization and foragers' activities, the most important factor that can induce social synchronization for light synchronization. After about 7 days under these conditions, we collected nurse and foragers and a control group of bees that were in the same room, but not in the hive. So they experienced only the light-dark illumination regime and not the colony environment. And we compared the locomotor activity as a reporter for their circadian phase. And here you can see the results. The nurse age bees in the cage. This is a circular statistic. So each one of these dots is showing the onset of activity for individual bee. Here it's the offset of activity. The dark phase is the dark part is when the hive was dark. This is when it was light. This is when the hive was open, and this is when the hive was closed. But these bees, as I mentioned, were in a cage. They were not related to the opening and closing of the colony. And you see that their onset of activity and offset of activity is locked very nicely on light on and light off. When we look on foragers, it's a little bit more complex. Let's start with the onset of activity. You see that it's associated with closing the hive and not with the time of light off. It's a little bit more complex for the onset of activity because it's probably affected both by opening the hive and by the light illumination regime. What about the nurses? They are the most interesting group here because they experience both the light and the forager activity. And as you can see, they are much more statistically more similar to the foragers than to the nurses, specifically related to the onset, which suggests that they are socially entrained, and the social entrainment is stronger than photic entrainment. And again, it was the first time that it was shown to any animal. Now, there is a possibility that the nurse bees can approach the hive entrance or being entrained by external factors. So we repeated this experiment with 2 additional colonies in which we caged some nest bees inside the colony and also monitor the locomotor activity. And then we found that basically they are very similar to the interface to the nurses and foragers and statistically different than the nurse HBs in a cage, showing that in honeybees, at least, social synchronization can be more potent than photic entrainment. Again, it was the first time it was shown in any animal. So if I summarize this part of the talk, our model is that foragers are entrained mostly by the environment outside, probably sunrise and sunset. They, in turn, can socially entrain the nest bees. They can also entrain each other. Probably the external environment has little effect, if any, on the nest bees. They can entrain each other. We don't know if they can affect the forager activity. And this is raising several important questions. One of them is what are the signals mediating social synchronization in the colony? And our hypothesis was that these are surrogates of forager activity. So it's kind of self-organized system in which when the foragers are active, they create some changes in the environment which synchronize the other bees in the colony. And we could think about several different factors like pheromones, temperature, comb vibration, and CO2, or other factors in the hive. We think the temperature is not very likely because the temperature in the center of the colony or the center of the hive is quite constant, and it cannot act as it cannot be an efficient time giver. But now I don't know if I do have time to continue or that it's too much. So we do have the time to hear a little bit about the other experiment, or that it's enough.
You can go on. You can finish. It's okay.
Okay, thanks. So to start asking what are actually the signals, Oliver Schiller performed several experiments. In the first experiment, he studied the hypothesis that the substrate-borne vibrations, when the bees are active, are entrained in other bees. So he took a group of foragers, put them in a cage, and in the same distance from them, he put newly young bees in cages. This was like 80 centimeter, either on the same substrate as we see here, or in a different substrate as we see here. And after 7 days for entrainment, he compared the phase of activity by analyzing their locomotor activity rhythms. And we use, in this experiment, we used the circular statistics to compare it. So in circular statistics, as I showed you before, each one of the dots is, in this case, it's the onset of individual bee. The foragers were entrained to a certain phase, which is marked by the bulb here. And the hive, which was free foraging, was entrained by the time of sunrise, which is shown here. And you can see that the foragers have a very long vector, which means that they are very well synchronized with each other. It's pointing to a time that is quite similar to the light on to which they were entrained. The bees on the same substrate are also pretty well synchronized with each other because they have a long vector, and they are more similar to the foragers than the bees on a different substrate. And you can see also that the vector here is shorter, and also if you look on the individual bees, that you can see that they are spread all over. So the bees on the different substrate are not as well synchronized with each other. And Over repeated this experiment with 5 different colonies. And you can see that the results are basically the same, very strong synchronization of the foragers and the bees and the young bees on the same substrate with a very similar phase. And he quantified it, and here you can see the vector length, which is a proxy for the degree of social synchronization. You can see that foragers and bees on the same substrate are very well synchronized with each other. The bees on a different substrate are less synchronized. And he also quantified the differences between the phase and the foragers in the phase of the bees on the same substrate and different substrate. And you see that those on the same substrate are much more similar to the foragers, which suggests that the foragers can entrain bees just by substrate-borne vibrations. In the second experiment, he compared the effect of colony odors, which is also a good hypothesis to underline social synchronization. So again, he put young bees in cages, and he had 2 hives. The experimental hive housed a free foraging colony with a little bit more than 1,000 bees. Actually, a little more than that, several thousand bees. And he sucked the air from this colony and put it over the cages of the young bees, so they were exposed to the odors of the colony. The control groups were connected to a similar system, but in the hive here outside the lab was similar, and it has the same number of honeycomb as in this colony, but there were no bees. We did heated this hive to the same temperatures to control for temperature fluctuations. And again, after 7 days for entrainment, he compared the activity rhythms. He already well familiar with circular plots, and you can see that the results were basically comparable. The degree of synchronization is much stronger for the bees that were exposed to the odors of the hive, which is consistent with the idea that the hive odors, we don't know exactly what are the hive odors. It can be maybe even CO2 or oxygen or other gases that vary during the day. Those are probably contributing to social synchronization. So the bottom line is that these 2 experiments show that both substrate-borne vibrations and colony volatiles can mediate social synchronization in honeybee colonies. To study this system with more details, we collaborated during my sabbatical in Washington University with several groups of engineers and biologists, and we developed a system to calculate coupling strength. So this system allows us, that we call this pipeline icon, allow us to measure the coupling between two individual bees or two individual oscillators. And basically these pictures, which I will not explain in details because we are a little bit short in time, is only the validation of the system that we published in 2018. And then now we use it to study social synchronization in bees. And in one of the experiments that I will show you, Oliver used this system to study synchronization of young bees by foragers. So we have these arrays of trays, and if each one of these circles here is a cage with an individual bees. In the open circles, he put a young bee. In the blue one, he put foragers, and he measured their coupling strengths between them and social synchronization between bees that are neighbors to foragers, farther away from a forager, or even farther away from the foragers, or even on a different tray. So the cages were connected with this tube, and it has a mesh, so the bees could antennate and could smell each other, but they cannot go from one side to the other. And it's allow Oliver to study the effect of direct contact. I've already showed you that bees can be entrained even without direct contact, but maybe direct contact nevertheless is also important. And we, for example, Oliver used this double plot actograms that I showed you before and compared the phase on day 2 and the phase on day 7. The bees were constantly monitored. And you can see, for example, for the foragers, there is very little movement. It's their internal rhythm is a little bit less than 24, so there is a little bit of difference. It's the same for young bees that are on a tray in which the cages are not connected. But if we look on a young bee that is connected to a forager, directly connected, we see that slowly they change their phase until it is similar to the phase of the forager. And if we summarize these results, we see in blue the foragers in day 2 and day 7, we see a little bit of movement. And these are 3 different colonies, 3 different trials. If we look on the young bees that are connected, we see this is in day 2 and this is in day 7. We see that they move to where the phase of the foragers. We see much less movement for the bees that are unconnected. It suggests, again, that the foragers can entrain the nurses and that direct contact is important. When we took the icon pipeline, we have a much more sensitive results. So this is the coupling strengths, which is showing how strongly one individual entrained the other. So here we can check pairs of bees. Circular statistics require a large sample. And you can see in the 3 different colonies that bees that are connected to each other are much better synchronized or have a stronger coupling strength. And actually, this algorithm allows us to check whether the effect of foragers on young bees and of young bees on foragers, and you see that it's working only in one direction. Basically, the foragers, which have strong rhythms, are entraining the younger bees. And this is like number 1 here. This is like direct contact, but the results were essentially the same when we look on the farther away neighbors and even farther away neighbors. Finally, we look on bees on the same tray or on a different tray, as we see here, and we see that bees that are on the same tray also have a stronger coupling strength, which suggests that, which is consistent with the fact that the subset vibration, even in this system, when we have only, on each one of these trays, we have only 30 bees. So, you know, it's very, very weak vibration, but still they are enough to entrain the other bees. We cannot detect this entrainment with the circular statistics, but with the coupling strength analysis, we can see it's a very weak coupling, but it's statistically significant. So to summarize my talk, so in the beginning, I was speaking about task-related plasticity in circadian rhythms, and we found that it's regulated by contact with the brood. It's detected by the antenna. I didn't show you the results, but we already published it a long time ago. And it affects the functioning of the circadian system. We don't fully yet understand what's happened with the circadian system, but we know that at least some of the circadian cells, the brain pacemakers, which are marked here by these circles with a cosine wave, at least some of them are cycling even in the brain of nurses. I showed you that at least most of the neurons, or not all of them, are cycling. There are some influences of the physiology of the bee that I didn't present today. And I showed you that we have studies that showing that even sealed brood that do not need to be fed can induce this plasticity in circadian rhythms. Then we ask about social synchronization, and we show that surrogates of activity like comb vibrations and colony orders can entrain circadian rhythms. So basically, bees entraining each other in a way that is very similar to these famous experiments with metronomes that you can find in many movies in YouTube, in which you put the metronomes, each one of them with a different phase, on some vibrating or moving substrate, and after a while they will entrain each other to exactly the same phase, and it seems that it's working for the bees in a very similar way. Finally, I want to thank the people that were involved in this line of research and other research in my lab and my funding agency and collaborators. The students that I mentioned during the talk, the students and postdocs that are underlined are those that performed the experiment that I showed you today. And I want to thank you again for inviting me and for listening for the talk.
Great. So we're going to do the questions right now. The questions will be sent in the Zoom chat also, so you can check them while I'm reading them to you. First, Patricia Silva asked, what is the method to make the bee stop foraging and start taking care of the brood again?
Okay, this is an excellent question. So the way that that is done is that the division of labor is socially regulated in bees, and if there is a shortage in nurse bees, some of the foragers will revert to take nursing activity. So basically, what we did is in this experiment, it can be done in several different ways, but what we did here is that we basically created the colonies with only foragers. So we collected foragers in the field, and they created these reverted colonies.
Great. So the next question is from Fernando Ribeiro Gomes. Does the juvenile hormone modulate circadian rhythm in bees?
That's an excellent question. So we tested it in both in honeybees and in bumblebees. And in honeybees, we didn't have any effect. And we published it, I think, when we didn't publish it, I think 2012, if I'm not mistaken. No, even earlier. And but we just published this year a study for bumblebees, and we found that there is an effect on circadian rhythms. And it's interesting because the other major line of one of the other major lines of research in my lab is related to these differences in the function of juvenile hormone in bumblebees and honeybees, and it's two different things. So in bumblebees, it's a gonadotropin, it's regulating reproduction. In honeybees, it is not affecting the ovaries, it's only affecting division of labor. So it's very interesting that the effect on circadian rhythms is also different.
Oh, that's very interesting. The next question is from Pedro Brizola. Sleep deprivation in foraging bees impair their efficiency in foraging. Do nurse bees have some sort of impairment as well?
This is about sleep deprivation, the question?
Yes.
Yeah. So that's a great question, because at least for bumblebees, again, we published last year a paper in Current Biology in which we showed that when they are exposed to the brood, they actually sleep less. And then there is a question, what are the effects? It's very difficult, I think, or at least it's not straightforward to study. Then we haven't done it yet. In honeybees, we didn't see a very clear similar effect that they are losing sleep.
Oh, nice. Oh, so the next question is from Danilo Flores. You see no rhythms in clock genes expression of nurses. This could arise from arrhythmicity in individual bees or desynchronization among bees. Have you tried to distinguish between these two?
Between the two, I'm not sure that I understand the question.
Okay, between arrhythmicity in individual bees or desynchronization among bees, like the collective.
Okay, excellent question. Yeah, this is a question of a chronobiology, someone that really understands chronobiology. Yeah, of course, it was our concern, and we cannot measure clock gene expression to individual bees, of course, but we can monitor locomotor activity for nurses that we remove from the hive, and this gives us a kind of a proxy for the phase of activity, and this is one way to see for individual bees. The other ways is to see the degree of variation between nurses. So if they are not synchronized with each other, there will be a lot of variation, and this is not the case. So it seems that the results are actually not because of lack of synchronization between nurses, and the second part of my talk actually showed that they are very well synchronized with each other by means of social synchronization. It's actually plasticity in the clock. But a related question that probably will be one of the next questions that you will ask me is that there is a discrepancy, at least an apparent discrepancy, between the results for the whole brain. When we analyze messenger RNA in the whole brain, there were no cycling, right? Yeah. Even in perioid. But when we analyze their expression in neurons, like the protein, in this case it's the protein level, nurses have very, very strong rhythms in pair expression in the clock cells, and it was the same phase as foragers. So we don't know. So our current working hypothesis that it's related to glia, as I told you, we analyzed a few glia cells in the analysis that we did before, but it was after quite a long time, and we don't fully trust these results. So this is basically one of the line of research that we are pursuing now. But it's a big question.
Yeah, it is. The next question is from Andrea Eleni. In leafcutter ants, a few million ants are organized in chambers that can be more superficial or are very deep in the soil. How to speculate about a model considering the emergence of rhythm patterns in a system like that?
So it's, first of all, as you mentioned, it's a system that is very difficult to study. One of my former postdoc, Tao Fuchikawa, he now has a position in Japan, and he tried to study termites, which is basically the same problem. It's there is no really good observation eye for termites or for leafcutter ants. But my hypothesis will be that it will be about the same. So I think that at least for systems in which the nurses and other nest bees actually stay in a dark and constantly regulated environment, I think that they will be active around the clock, because I think that activity around the clock is beneficial for them. They can provide better care for the brood. So, you know, that when we want to have a fish or, for example, chicken, when we want them to grow really fast, we provide them food ad libitum and we illuminate their cage throughout the time, and then they just eat more and they grow faster. So we believe that this is the case, but just by means of natural selection in this case, in social insects. So when nurses are active around the clock, they can rear the brood faster, and the colony can grow faster, and this is it's functionally significant. It's improved the colony fitness.
Yeah, great. The next question is from Pedro Brizola. He says, I've read that foragers bees from different petri lines have a different preferential foraging shift. What could be the mechanisms underlying this pattern? And what could be the consequence of this pattern to the colony synchronization?
Yeah, this is interesting, and I've seen this results, and there are also recent study suggests that the bees can be entrained by their different flowers. So there are like cohort of more morning active bees and evening active bees. We never really studied it explicitly, but we didn't see it in our colonies, because when we collect foragers, we don't know exactly if they are going to early flowers or late flowers. So our colonies are free foraging, and probably they have many sources, and they show quite good rhythms. I think that there is only one study about the genetic differences, which is it's genetic basis. Yeah, it's interesting question. You know, we know from studies with model organisms like mice and Drosophila specifically, that there can be variability or natural variation in clock genes, which can affect properties of the clock. So there are, for example, shorter or longer rhythms, and in human, we even speak about this chronotype, the owls and the larks, you know, like early and late chronotype. So there is no reason to assume that bees cannot have these natural chronotypes, because we assume that natural variation will work on properties of the clock. Now, how exactly is this variation is contributed to the function of the colony? I don't really know, but it's theoretically it can contribute.
Yeah, great. So the next question is from Natalia Araújo. Any thoughts about how the molecular clock changes according to the season, summer, winter? And she also asks, the intensity of the sun radiation is as important as the number of sunlight hours?
Yeah, it's again an excellent question, which hint for me that many people in your group are actually have extensive training in chronobiology.
Yeah.
So one thing that we need to understand about the bee system is that it's in many ways different than most of the other animals that probably you study. So at least the group of audit that I know would study. Actually, some of the organisms are similar, but when we speak about cavity-dwelling animals, like cavity-dwelling rodents that I know from some of the studies from your group, and bees, basically they determine the light-dark illumination regime. So when a bee or a rodent decides to go out of the tunnel or the colony or the nest, they are exposed to light, and they basically determine their light-dark illumination regime. It's not imposed of them like on Drosophila. So this is one aspect. You know, the intensity of the light, again, it's it can create stronger photic entrainment, which might be to some extent important for the foragers. But I think that even during the winter, I think that light is definitely strong enough to entrain their clock. But we did do one very small study about seasonal factors, because it's interesting, and I did it when I was in Illinois, and we did find evidence that even in bees that can so nicely control their environment, there are seasonal effects on the clock. So the free running period and the age in which they show circadian rhythms, these are all a lab experiment. So we collected bees from colonies around the year. Again, it was done in Illinois, in which there are strong seasonal fluctuations. In Israel and definitely in Brazil, it is not as strong. And we did have evidence, and it was a very small study that we published, I think, in 2008. And we never extended. We just published it, hoping that additional people will take this line of research and extend it.
Yeah. Great. So the next question is from Arthur Roque Justino. He asks, are the mechanisms responsible for circadian rhythms in honeybees similar to different social insects?
So now we have quite a few ant and even wasps genomes. And this mechanism, if I understand the question correctly, it relates to the fact that the clock is working with a mammalian type cry, what we call cry M, and without timeless. So it seems to be the system in all the Hymenoptera. It is different in termites, which are social, and of course in other social animals. So in the beginning, we thought that maybe it relates to sociality, but when they actually sequenced the Nasonia genome, which is a solitary wasp, they found that it's the same system. So we think that this is typical to the entire Hymenoptera. Now the question is where another facet of this question is whether social evolution, whether we can track some selection pressure on the clock genes in social insects. We were thinking many times on pursuing this line of research. Actually, we never get to do it. But I'm not sure that this will be, I think that this will be very difficult to find it, because I think that most of the regulation is above the level of the individual clock genes, because the same clock genes can move, for example, in honeybees. Foragers have really extremely strong circadian rhythms, and they use it, as I showed you in the neuroanatomy. It's actually, at least when we look on PDF, it's innervating, very extensive innervation of the entire brain, and we know that the PDF fibers are actually contacting many brain centers that control, like sun compass, for example. So they have, on one end, a very strong clock. On the other end, it can be weak. So it's not, I don't think that it's in the level of the clock genes themselves. It's some higher level that is controlling plasticity. And of course, we know nothing about the endocrine or other regulation of this plasticity. We only know about the behavioral and social regulation.
Yeah. Okay, so the next question is from Gisele Yoda. She asks, would the queen have any coordinating role in the timing of the colony?
Yeah, we have very high, so there is a paper that actually argued this to happen, and they actually transfer queens from different colonies, and they argue that it's affected the phase of the colony, but they didn't have the same methods that we can use now. we and others did the experiments, and we now have very high resolution experiment from monitoring from observation types of tagged queens that we monitor them with a barcode monitoring system. And the queens are active around the clock, they have no rhythm. And they have, you know, that honeybee queens, they have very strong negative phototaxis. So basically, if there is light, immediately they escape from light. So they stay in basically in the what we call the brood nest, which is the area that is best thermoregulated. The environment are most constant. The queens are laying eggs. Now we have a paper that we are preparing for publication in which we show that the queens are actually laying eggs around the clock. And so they are not exposed to the external environment. They are active around the clock. I don't see any way in which they can entrain the other individuals. But it's, you know, we cannot prove that it doesn't happen, but I don't think that this is the most important. I think that it's really like a self-organization system like the one that I showed you with, which is the driving force is the activity of foragers. So foragers compose like 35% of the population of the colony. They have very strong rhythms, and they create these oscillations in the environment that we are just starting to characterize.
Great. So the next questions are from Leigh. Did you test cycles in reproductive workers and compare gene expression with queen bees?
So again, it's depending if we ask about bumblebees or honeybees. So in honeybees, workers will hardly reproduce. In order for the workers to reproduce, we need to have a queenless colony without the queen. And it's also depending on if there is a brooder, they will be active around the clock and they will not be reproducing. So, but the short answer is that we never tested the reproductive workers. Again, in bumblebees, it's a little bit different because the workers typically reproduce. So the regulation of reproduction is not very effective, at least at a certain stage of the colony. And many workers lay eggs, and this is related to juvenile hormone. So in bumblebees, there is actually an interesting issue. So the one thing that we can say that the bumblebee workers and bumblebee queens, if they are exposed to brood, they will be active around the clock similarly, and if we remove them from the brood, they will develop rhythms. But other than that, we don't really know.
Okay. The next question is, do paternal line influence degree of synchronization?
We never tested it.
What? Can you?
We never actually tested it. So
yeah.
I would just mention that for some of the experiments, specifically when I just started working on this system, we actually use what we call SDI colony, single drone inseminated queens, to reduce as much as possible the genetic variation. And we thought that it will improve our statistical power because we analyze clock gene expression for only, we want to analyze for as fewer individuals as possible because it is expensive. But actually we discovered that it has no effect. So now we are using naturally mated queens, and the sample size is not enough. It needs to be a huge experiment in order to really test it, at least in the molecular level. In the behavioral level, it's more important, but we never tested it.
Yeah. Okay. And the last question is, we have another 2 questions actually. The next question is, immature do not exhibit cycles?
Again, I was reading the chat. There was a question. Sorry for interrupting because there was a chat and I was reading it. Do virgin queens have a rhythm?
Yeah,
So
these are the next two questions.
are the same that you're seeing. But anyway, virgin queens have very strong rhythms. It's a great question. And actually, studies that were done in Asia, in which they have several species of honeybees, you know, in Brazil and in Israel, we have only one species, which is the European honeybee, or the Western honeybee, Apis mellifera. In Asia, they have several species, and actually the virgin queens have different phases, which probably help them to separate their mating flights. And it was shown to be the same for ants. So again, this is the plasticity that I mentioned, that there is a reproductive related plasticity in circadian rhythms that I didn't show you the results. In this case, virgin queens have very strong rhythms, and egg-laying queens have no rhythms. They are active around the clock, at least behaviorally. And we never look into the brain, just because it's more difficult to work with them. So now I didn't hear your question, so can you repeat it?
Okay, sure. The last question is, immature do not exhibit cycles?
Again?
Immature do not exhibit cycles. I believe it's about the immature bees.
The young bees?
Young bees, yeah, I think it's about the young bees. They ask if they do not have any rhythm patterns.
So I don't know if the question relates to individual. So if we take individual, let's see if my answer is actually addressing the question. But if we take individual bees, individual newly emerged bees, and in many of the experiments, that's what we are doing. We take a newly emerged bees just after emerging from the pupa, and we put it in individual cage. In both honeybees and bumblebees, typically in the first few days, they will not show rhythm. So they will show a development of rhythm. And this is when they are entirely isolated individually. And there is a significant genetic variation. So different colonies will show a different age in which they develop rhythms. It can be in a few days, like 3-4, and it can be like 20 days. It's also related to their ability to function normally in isolation. So you have to remember that being individually isolated is probably a very stressful condition for any animal, but specifically for social animals. But we use it, we still can learn something, but differences between colonies can be related to many factors, not only those that determine circadian rhythms.
Okay, so the next question is from Maria Cristina Arias. She asks, if the foragers determine the rhythm in the colony, what do you think happens to the solitary bees?
I think that solitary bees are more similar to other solitary insects, but some of them have like a kind of subsocial behavior that they do care for the brood. So in this case, they might need to be active to some extent. In most of the solitary bees, they actually provision cells. They put pollen and nectar. We call it a bee bread, and they lay the eggs on top of it. So I don't think that they are. And then the larvae just emerge inside the food, and they just can take care of themselves. So I don't think that that it's really the phenomena. But one interesting question that actually relates to Brazil, and maybe I think that even some people from your university were discussing it with me, is what happened with stingless bees. Because with stingless bees, they have this cell provisioning. So the nurses are totally different than nurses in honeybees and bumblebees. And I don't know if any one of you have a good answer, but I don't think that it's exactly the same. the foragers definitely have very strong rhythms. Probably the queens are active around the clock, but the workers, I don't know exactly how it's worked with the stimulus. Anyone knows? Here?
Yeah, not sure. Okay, so I think that was, oh, we have another question from Lays again. One more. Vibrations are perceived through the antenna when you talk about the synchronization.
The antenna has multiple functions here. So we have a paper in regard to plasticity. So there are two systems that I showed you. One of them is plasticity in circadian rhythms, is what is causing, what are the social factors that are causing to an individual bee to switch between activity with and without circadian rhythms. And what we showed is that if we remove the antenna, we couldn't block them, so we need to remove them. They will have actually strong rhythms. And when we analyze them individually, they have no rhythms. It didn't affect the rhythm. So the removal of the antenna doesn't affect circadian rhythms of the bee when they are individually isolated. But when we put them in the colony, and they are supposed, it's nurses that are supposed to be active around the clock, they actually show rhythm. So this is showing that the antenna are necessary to perceive the signals that induce bees to switch between activity with and without circadian rhythms. We don't know if this is the only sensory organ, because they can also sense it with their maxillary pulps and taste, and maybe even legs. They can sense some chemicals, but the antenna is at least one of the factors that is important. Now there is also the issue of the phase of the antenna. So I didn't show you the results, but the antenna is a cycling in bees as in other insects. And the cycling are a little bit different between nurses and foragers. And then the question is, what is exactly entraining the antenna? So I gave a more elaborated answer, and we don't know if it's by chemicals or by some neuronal pathways that are different between nurses and foragers.
Okay. We have finished the questions. So I think we can finish the session right now. Thank you very much once again, Guy, for giving us such an amazing talk and an amazing discussion. I would also like to thank everyone who watches us today. I hope you enjoyed this transmission, and we'll be waiting for you again on our next Physio webinars. Thank you.
Thank you very much. It was a pleasure.
Okay, we're